Hello, it's a beautiful day here in Laramie and a perfect time to talk about translation of messenger RNAs to make the protein product. So recall that our adventure has entailed looking at the processes of replication and then most recently transcription, whereby in transcription the DNA is used to encode for the RNA, and in the case of messenger RNA or mRNA, we know that that carries the transient code that encodes for the production of a protein product. So our goal today is to look at that process of converting the mRNA code into amino acid code as a part of the protein product. So all of you had the challenge last time in your activities of looking at the DNA coding strand for a particular gene encoding for an antimicrobial peptide. This peptide called acidosin is a peptide produced by lactic acid bacteria. It allows them to compete for resources in space in these fermented products that we like to eat. So it's an important antimicrobial secretion by certain lactic acid bacteria. So this particular gene sequence was the one that we looked at, and we took note that this particular G was the +1 start site for transcription. So it was at that site that transcription would begin, and that we would see that the protein, or excuse me, the RNA, the mRNA product would begin being made at that first +1 start site. But note that this is the DNA coding strand. So this is the strand that gets up and out of the way while RNA polymerase reads the template strand to make the RNA. So when the template strand is making the RNA, that means that the RNA is going to look pretty much identical to this DNA coding strand, only we know that UTPs, or excuse me, we know that NTPs rather than DNTPs are going to be used to build it, and also that every time we see a T, that would be replaced by a U in the mRNA. So this was one of the answers given by one of your teams on the discussion activity string, and you noted that the mRNA that would be encoded for by this DNA would in fact have that same sequence, G, C, only every time we see a T, that would become a U. So we can ask that question, do we agree? I don't know. You'll have to look back at your discussion threads and see if your teams also agreed that this would be the mRNA sequence. So when we look at an mRNA, this mRNA has multiple regions on it, and not every one of these regions is going to get converted into protein product. In fact, only the center coding region that I've shown in red here will be translated. That means that upstream of the translated region is a non-translated leader sequence. Downstream of the translated region is a non-translated trailer region. We can hypothesize the reasons for having those non-coding sections. For example, picture a Pac-Man-like enzyme that is a 3' nuclease that chews from the 3' end here of the mRNA, chowing down on it. So this non-translated trailer region is a great protector. It's like armor to the coding region. The non-translated leader sequence can protect as well, but it also enhances translation. So it provides a landing pad for the translation machinery. And specifically, we know in that region is going to be a site to which the ribosome is going to bind. So a ribosome binding site. When an mRNA is read to make a protein, we have something that we need to think through. Remember that mRNAs are made from a sum total of 4 nucleotides: UTP, GTP, CTP, and what am I missing? ATP, right? So we recognize that those are the building blocks. There's only 4 of the nucleotides in any given mRNA. And yet, the mRNA somehow manages to code for 20 different standard amino acids. So how is it that 4 nucleotides can code for 20 different amino acids? It wouldn't work out. If each nucleotide encoded for 1 amino acid, there could only be 4 amino acids. So it must not be that 1 nucleotide encodes for 1 amino acid. It must instead be that combinations of nucleotides encode for amino acids. So there are 4 nucleotides that comprise RNA. And so if those were read in a combination of 2, that would be 4 squared, or 16 possible combination. That still falls short of what is needed because there are 20 standard amino acids. And so therefore, the RNA must be read in triplicates. These triplicates are called codons, and there are 64 of them. What that means then is that that must necessitate there being many more than 1 nucleotide triplicate that encodes for the same amino acid. So said in another way, the genetic code is degenerate. There is more than 1 codon that encodes for a single amino acid. The codon usage table shows this degeneracy. You can see that, for example, the codon CCU, CCC, CCA, and CCG all encode for a kinky amino acid proline. So it's cool to see the location of degeneracy. Recognize that it's always in the most 3' position of the codon that we see that the change is allowed. So it's like the loosey-goosey position of a codon is that more 3' position within the codon. So even in some areas where we don't see that 4 codons encode for a single amino acid, we might see that 2 similar sets of codons might encode for similar amino acids. So for example, if we were to say look at glutamate and aspartate, our 2 superacidic amino acids, we can recognize that GAU, GAC are aspartate, but then change just that more 3' nucleotide, GAA and GAG, and glutamate is the amino acid that gets incorporated. So similar amino acids also may be a part of this sort of degeneracy in the code. I want to draw your attention first to the usage of the table and then some other special codons. So if we were trying to figure out which amino acid would pair with the codon CGU, how would we do that with this codon usage table? Well, we'd start with the left is the first position, so C, the top is the second position, G, and the right is the third position, U. So hey, it's our good old friend, the ARG, the pirate amino acid, the most base of them all. CGU encodes for arginine. So this is how you use the codon usage table. I also want to draw your attention to the 3 stop codons. When UAA or UAG or UGA are encountered, that essentially triggers the ribosome to stop incorporating new amino acids. AUG, on the flip side, is the start codon. So AUG will encode with a special amino acid, in fact it's always methionine, or a formulated derivative of methionine in bacteria called FMET that is incorporated every time that first AUG is encountered. Now, of course, it has to be the first AUG that is downstream of the ribosome binding site, because then it has to be the AUG that occurs after the ribosome has bound and initiated translation. So speaking of the ribosome, let's check it out. Remember that in bacteria, the ribosome is a 70S. It has a 30S and a 50S subunit. One's tempted to think that the 30S is always together with the 50S, you know, like they're socially isolating together and they're always together, you know, but in fact they're actually socially isolated. They actually spend time away from one another and they only come together to do the job of translation. The 50S subunit is comprised of a 5S and 23S ribosomal RNA and 34 polypeptide chains. The 30S subunit has the epically famous 16S ribosomal RNA and 21 polypeptide chains. When I say that 16S is epically famous, well, part of that is because of us. We have sequenced 16S rRNAs to look at evolutionary relatedness and identification of bacteria. The 23S rRNA would probably argue pretty vehemently with me that it should be more famous because it's catalytic. It's actually the ribosome that forms the peptide bonds in incoming amino acids as the polypeptide chain is built. So in that way, maybe it would argue that it should be the most famous. Ribosomes are one important player in the process of translation, but tRNAs are equally as important. tRNAs, as Francis Crick termed them, are adapters, and that's a great term for them. Francis Crick is a little bit interesting, but this was a really good contribution that he made in thinking about tRNAs and the way they adapt. So a tRNA has multiple regions. It has a T-C arm, a variable arm, a D arm, but most importantly, the anti-codon arm is the sequence of nucleotides on that anti-codon arm is what recognizes the codon and therefore enables it to pair with its appropriate amino acid. So if we go back to our codon usage table, let's check it out, and remember that the codon that was getting recognized was AG. Well, that tRNA is therefore going to carry lysine and pair that codon with lysine. So notice that on the acceptor stem of the tRNA, lysine is the amino acid that is carried on that tRNA. So the tRNA is adapting the codon to the amino acid and allowing for that pairing to take place. As you might have guessed, real quick before I leave this behind, make sure you understand how you would look at an anti-codon sequence and pair it with a codon. Recognize that it has to be anti-parallel with the codon. So if the codon is 5' AAG, therefore the anti-codon must be the 3' end, 3' UUC, 5', right? It must be anti-parallel to the codon. Now, in three-dimensional space, a tRNA actually wraps up and takes on a 3D kind of shape. It almost looks like an L or a liver kind of shape. It's 3' hydroxyl. There it is, the acceptor stem. That's when an amino acid is activated and ready to be paired with a codon via the anti-codon. That amino acid will be attached to that 3' acceptor stem. So tRNA, really, they are adapters. So let's take a moment to look at a video that one of my students made helping him understand tRNA.
I was struggling to come up with a way for me to understand how adapters work. The tRNA adapters that go between the mRNA codon and help connect amino acids. So I was trying to think of a visual way to understand this, and I thought of RF connectors. Anytime you make a cell phone call, it's thousands of these which hook together and allow that to happen. So what's in front of you here is an mRNA. And let's pretend that each one of these represents a codon. They're a little bit different, and they each have a different task. So if I take an amino acid, let's pretend that this is alanine right here in my hand. Alanine needs to fit on here, and it can't. You can see that it just doesn't work, but it has to. So to the rescue will come an adapter. An adapter allows for the alanine to connect to the codon in a way that will allow the data to transfer appropriately. Pretty simple, right? Now, because there are 61 distinct tRNA species, but 31 if we take into account the wobble effect, there are different tRNAs. And though they're a little bit different, the same in shape, this video kind of shows them a little bit differently. So over here we have another one. We try to fit on our amino acid, and it simply won't work. They're the same thing. So, again, to the rescue will come our adapter.
let's go ahead and practice a question to see if we can make good tRNAs and recognize how a tRNA anticodon sequence will enable us to recognize what amino acid is paired with a codon, and to see if this particular anticodon sequence in this question, ACU, will carry which amino acid on its acceptor stem. So we want to figure out what the answer is to that question, and then does this tRNA play a role in reading the mRNA whose sequence we wrote in Friday's activities? Let's see if we can secondly answer that question. So let's start by thinking about the tRNA and its anticodon sequence. Remember that if a sequence is just written like this without any termini specified on it, we have to make the assumption that we're being given the 5' to 3' sequence, so that this is the 5' end. That 5 is epic, but we're going to go with it because this pen is not very responsive. So if it's 5' ACU, and we know that a anticodon is always anti-parallel to the codon, and codons are read 5' to 3'. This then must describe the anticodon arm, right, would be, if I'm just drawing the anticodon arm and the rest of the tRNAs up here and up here, but this is the anticodon arm, and we know it goes 3' to 5'. This would have to be U, CA, as it situates on the actual anticodon arm. And then that's going to base pair with the mRNA. So I'm going to try to see if I can make the mRNA a different color. We'll make the mRNA blue, so we know then that it's going to be going in the 5' to 3' direction. So the codon that UCA would recognize, U will pair with A, and then C will pair with G. And then finally, A would pair with U. So AGU would be our codon on the mRNA. So that nice anti-parallel orientation of the anticodon and the codon. So then the question becomes, in the mRNA that we've already written for our peptide, do we see the codon AGU occurring at any place on this mRNA in the coding sequence? So that means in order to find the coding sequence, the region that is actually converted into polypeptide chain, the first thing we have to hunt down is the AUG start codon. So we recognize that this whole region right here is the 5' untranslated region, and we need to scan down this looking for the first AUG to know that that's where the reading frame will start, where we will set the triplicates to read the codons. So I'm just going to look for anywhere I see AU. So here's AUA, AUU, AUU, AUA, 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 AUG. Look at that. There is the start codon. So then that sets the reading frame in triplicates. So AGU, check it out. It is literally the second codon. So this tRNA with an anticodon sequence of ACU will recognize the second codon in our mRNA. So then now what you'll have to do to find out which amino acid is connected way up above there on the acceptor stem of that tRNA, you'll need to use your codon usage table and look up AGU to find out what tRNA that, or what amino acid that tRNA carries. So as we remember in the anticodon, or in the codons that we looked at when we looked at the codon usage table, remember that that third position was this point where there was all sorts of loosey-goosiness as to the amino acid that was encoded for by that codon. So remember, we could see like 3 different or 4 different codons that would encode for the same amino acid, and the one place they changed was in the most 3' position of the codon. Well, that means that the most 5' nucleotide of the anticodon must be something special. It must mean that that most 5' nucleotide must literally be flexible in its capacity to pair with the most 3' nucleotide of the codon. So that's a special nucleotide, and it's actually capable of something called wobble. So this phenomenon that doesn't sound very technical is actually a technical term, that the most 5' position of the tRNA is capable of a wobble that allows it to loosely pair and recognize. This is actually, if you want to get really sexy minded, you can say to someone, guess what? Wobble is what allows degeneracy, because it is the wobble in that most 5' nucleotide of the anticodon that allows the flexibility in the 3' nucleotide of the codon in allowing it to pair with the same amino acid. So if we see a tRNA that has a C in its 5' nucleotide, it can pair with just a G. But check it out, if there's a G in that 5' position, it can pair with either a U or a C. So if we had an anticodon, GUU, which of the codons would it pair with, and which amino acid could it carry? So let's think about what would be true. If that is GUU, 5' to 3', we know it is UUG, so let's draw that out. That on the anticodon arm, just always when in doubt, draw it out. So this is a pretty ugly anticodon arm, but say that this is the anticodon arm, and it's U, U, G. Well, that's about the lousiest G I've ever done. And that would then, we know, would pair with oh, wait a minute. So there's a G, right, in the most 5' position of the anticodon. So it could either pair with a U or a C. So it would be, the codon would be A, and then that last position, or the most 3' position of the codon, that G could either pair with a U or a C, so it could be AAU, or it could be AAC. So this is kind of cool, right? Because it means that while there are, that is a C, while there are 64 codons, there are not 64 tRNAs because of this wobble effect. In fact, there's about 40 tRNAs to recognize those 64 codons. So let's see if we can go look this up in our codon usage table. There it is. So A, C, or AAU, both encode for asparagine. Excellent. That's great. So the amide derivative there of aspartate. So great. So we see how two different codons could be recognized by the same anticodon on the same tRNA carrying the same amino acid. Awesome. So then you can answer this question, is this anticodon involved in the translation of the mRNA that we wrote on Friday? And again, I'll leave that one to you to explore. So let's talk about the phases of translation as they take place. There are three phases. There's the initiation, the elongation, and the termination. When initiation takes place, we know that the first thing that's going to have to happen is that in the 5' untranslated region of the mRNA, this is where the ribosome is going to bind. But guess what? As we talked about, there's a lot of social distancing between the 30S and the 50S ribosomal subunit. So it's only the 30S ribosomal subunit that recognizes the Shine-Delgarno sequence, or otherwise known as the ribosome binding site, on the mRNA. So it actually pairs with that. Now you might be remembering that inside of that 30S subunit is the 16S ribosomal RNA. And it's the 16S ribosomal RNA that is pairing with the ribosome binding site. I'm going to show you a more in detail picture of that in just a moment. There are other things that get involved with this process of initiation. Of course, we know there's going to be the tRNA that carries the formulated methionine that pairs with the AUG start codon. So this little pink region here, that's the AUG start codon. But guess what? IF1 actually prevents that tRNA from binding unless it's bound to the initiation factor 2. So IF1, IF2, and IF3 are all initiation factors that are required for initiation of translation. IF2 is kind of like, I think about it as being like the adapter to allow that FMET carrying tRNA to bind to the AUG start codon. IF2 also has a high energy GTP bound to it, so that can be hydrolyzed to power the initiation phase of translation. Translation is an energy hogging process. I mean, a big majority of the cell's energy goes to translating. It's absolutely huge. So the other initiation factors coordinate all of this, preventing the 50S subunit from binding prematurely and enhancing the binding of the 30S subunit, and so enabling this process to take place. After initiation has occurred, of course, we know that the thing that has to happen next is going to be elongation. But before we dive into that, I promised you that I would show you a zoom in picture. So if we picture we're zooming in right here, where we're going to see the sequences on the ribosome binding site here in the blue, we're going to see what that sequence is. And then we're going to see how the 16S ribosomal RNA is actually binding with, or base pairing with, that ribosome binding site. So if we zoom in right on the 30S subunit right here, inside of that 30S subunit is the 16S ribosomal RNA, and it's going to pair complementarily with the ribosome binding site, or the Shine-Delgarno site. So whereas this is a purine-rich site in the mRNA, it's a pyrimidine-rich site in the 16S ribosomal RNA. So it is that 16S RNA that says, whoa, hey, start translation right here. So now we can move into elongation. Once the GTP is hydrolyzed, the initiation factors get the heck out of Dodge and the 50S subunit binds. Now the process of each tRNA binding to its codon, and the anticodon pairing, and carrying the peptide that will be added to the polypeptide chain.